Hello students, I Asanoja welcomes you all in the another lecture of my video session. Friends, by the time we have studied, or I should say we are studying unit number 6 of your biology chapter, and we are discussing chapter 2nd. If we talk about this chapter, till so far, we have studied all the concepts which involve in sexual reproduction of flowering plants. And today is the last lecture of this chapter. In this lecture, we are going to complete or cover or study two important topics. The first one is seed formation and second one is fruit formation. Friends, this both is collectively termed as seed and fruit formation. So, without wasting time, let us start. By the time, these are the three events. This one slide I am showing you from all the in all the lectures, and the basic concept behind that is you should not lose the continuity of the topic. You will happy to know that whenever it is asked to us that who is going to take the class tomorrow or day after tomorrow, all your teachers are so eagerly waiting for the moment and they fill the sheet very fastly. So it is hardly to get the time for our students, but still and your responses, your time responses are definitely too important, and you all are very perfect in that. So congratulations for all of you. Okay, so this topic we have discussed. The first one is pre-fertilization stage. All the events of gametogenesis we have studied here. The second one is double fertilization stage, how the fertilization takes place in angiosperm, and the third topic of post-fertilization event we are discussing. In my last lecture, we have discussed about its first part, that is embryogenesis. In the previous video, we have studied about the embryo formation, how embryo formation takes place in dipart, how it takes place in monopart. And we have also discussed about the detailed structure of embryo. And today, we are going to deal with the next part, that is seed formation and fruit formation, along with some of the terms which are given in your NCERT and which are very important as far as board examination is concerned. So, let's begin the topic. One diagram I have shown you in my previous video lecture, in which we have compared an embryo sac before fertilization and after fertilization. The embryo sac get changes. Similarly, the whole ovary get changes after fertilization. Once the double fertilization takes place, in which one male gamete fuses with egg to form a diploid zygote, another male gamete fuses with polar cell to form a triploid endosperm, and then the scenario of this ovary get changes. So let us discuss about the various parts of ovary which is before fertilization and what happened to after fertilization. And this will be the beginning of the topic for seed and fruit formation. So, the first one is ovary. As far as we are talking about the ovary is completely converted into the very important structure that is called as fruit. Hence, fruit may also be defined as a ripened ovary. Ripened ovary. After fertilization, again the changes takes place, the accumulation of endosperm takes place, the formation of zygote or embryo takes place, and this all complete structure, what fruit you are eating, is nothing more than a ripened ovary. And that's why in angiospermic plant, flower is a sexually reproducing part and fruit is the result of a sexual reproduction. Okay. Next one, the ovule. Ovule get converts into the seed. When once the fertilization takes place, the seed get converts into the ovule get converts into the seed. Okay, so remember that one ovule which is present there, so that converts into the seed. This seed is, as in the previous topic we have studied, today also we will put some more light on that. It may be endospermic, it may be non-endospermic, albuminous and ex-albuminous seed. Remember, we have discussed in our previous lecture. The wall of ovary. Now if you all remember, this is the ovary, as here is the embryo sac and this is the wall. The wall of ovary that converts into the wall of fruit, and which is called as pericarp. Pericarp, also called as wall of fruit. So remember the mango, it is a season of mango. Most of you are taking the delicious tastes of mango. So whatever you peel out, that is nothing but the outer wall, and that is called as pericarp. Next, in the ovary, whatever integuments which are present here, these integuments are converted into the seed coat. Remember, we have discussed about unitegmic, bitegmic and all. So on the basis of the integument after fertilization, it converts into the seed coat. And here in bitegmic ovary, we have studied that there were two layers, external and internal. Outer integument and inner integument. For such ovaries, outer integument converts into testa. A very important point as far as your examination is concerned. The outer integument of ovary converts into a thick, tough coat called as testa. And the inner integument converts into the inner layer, that is called as tegument. Tegument, inner layer, testa, outer layer. The other part is nucellus. Again, if you see this structure, this is the embryo sac. This is the ovary, this is the embryo sac, and this embryo sac is filled with various nutritive tissues. These nutritive tissues were called as nucellus, and this nucellus either completely degenerate. Actually, in most of the cases, it completely degenerates because maybe it's a development of embryo, maybe it's a development of endosperm. Remember, endosperm during development require nutrition which it gets from nucellus. So this nucellus is completely absorbed, but in some of the cases, it does not so. In some of the cases, the remnant of nucellus persists, remains, and that is called as perisperm. Okay, so either the nucellus is completely degenerates or it remains in the form of perisperm. These all terms we are going to discuss today in a much detail. Next one is egg cell, that you know, one cell. After fertilization it converts to zygote, and zygote again undergoes multicellular division to form embryo. Zygote modifies into embryo. The polar cell modifies into endosperm. I think these two parts we have discussed in a very elaborate way in my previous video. So you know about that. Synergids and antipodal cells. Synergids. Remember, what is the function of synergids? It is used for guidance of the pollen tube. Now, after fertilization, there is no use of the synergids. And during double fertilization, I told you also that as the pollen tube enters in embryo sac, the synergids get degenerates. So antipodal cell and synergids, they both undergoes degeneration, and it degenerates and lost. Next, micropyle. We have again studied about two ends, micropylar end and charazal end. So micropyle, what is the micropyle? Micro, small, pyle, mouth. That is a small opening, and this small opening, micropyle, remains as a small opening of micropyle of seeds. So the micropyle remain as an open structure. In a very small open structure, it is present in the seed. What is its function? During seed germination, it is the part from where the oxygen, gases and water, these all things get exchanged. And the last structure, that is funiculus, that is funicle. Remember, there was a stalk-like structure, and this is stalk-like structure only, then it converts into, here various embryos sac is there. This is called as funicle. This funicle remains as the stalk of the seed. So whatever the stalk seeds are there, it is nothing but it is the mature or modified funicle. Okay, so these all are some of the important parts of ovary or of gynecium before fertilization and after fertilization. For 1 mark, these topics are very, very important. Now, if we see a structure of a typical seed, at this moment of time I don't want to compare it or distinguish between monocot and dicot seed because monocot and dicot embryo, the structure of monocot and dicot embryo we have already studied. Okay, so I don't want to go in that part, but a typical seed is appearing something like this. Now, for some time, it is actually bean seed, but sometimes don't think it is a monocot or dicot seed. Just forget it. Okay, a typical seed. So, what is the seed? It is the final product of sexual reproduction. Again, very important question for 1 mark. Okay, during the sexual reproduction, seed formation takes place during the sexual reproduction. And what is the ultimate goal of the sexual reproduction? Is the formation of a seed. And this seed later on develops or grow into a plant or whatsoever. Another one, seed, if I find this line, that's why I have included here. If in one line I have to define what is a seed, so I can say that seed is a mature integumented megaspore. It is a mature, it is integumented. Integumented means covering. Megasporium, whatever megaspore is there, that megaspore converts into the seed. So that is one very important line I got included here, is the mature integumented megaspore, right? And if we talk about the seed is typically composed of 3 important structures. The first one is seed coat, external covering, seed coat. As you can see here, the first important structure is seed coat. Okay, this seed coat, also termed as an testa, that is the outermost covering. Okay, it play a very important role for maintaining the viability of a seed. In today's topic, we will see also what is the significance of seed, right? And you are very surprised to know that seed can maintain its viability up to 1000 years. It can maintain its viability up to 1000 years. Okay, this benefit is only with the higher plants. If we talk about bryophytes and thallophytans, they don't have seed formation. So whatever the result is formed, if that result does not grow at that moment, so that will lose its viability. But in angiosperms, it is there. If you remember, normally what we are cultivating the rice or grain or whatever it is. So whatever grain is there, you must have seen that farmers keep some grain. And these grain will be used as the as a seed for the next cultivation. Why so? Because seeds after 1 year also they have their viability. If you take the seed, sow it off, you purchase the gram. A very simple example. You purchase grams. When the gram was cultivated, when it was formed, nobody knows. But after 1 year also, if you put it into the water, it starts sprouting. What does this mean? This means that seed has its viability, and this viability, it has a tendency, and this tendency to grow into the plant remains in the seed for long years. Okay, and that is due to the seed coat, external covering, which prevent the seed from any adverse external conditions. First part. Second very important part is embryo. So if I am talking about the seed here, you can see this is the embryo. A very small structure, which can be seen here, that is the embryo. This embryo has plumule, radicle. Plumule to radicle is called as a shoot, embryonal axis we have studied about. So if we talk about the embryonal axis, this is the plumule, this is the radicle. So from here to here, this is the embryonal axis. This is the embryonal axis from here to here. Okay. So this is the embryo, which later on forms the new plant. Right. The another very important part. This is embryo is the second part. For regarding embryo, we have discussed in elaborate way in my previous video. The third one is storage of food or food storage. Food storage is nothing but in the form of a nutritive food storage. So whenever this embryo grows, this embryo required nutrition. And from where it will take the nutrition? Remember, we have discussed about cotyledons. Okay, so cotyledons were there, and this is in monocot it is called as a scutellum, and dicot we have two cotyledons. So that forms the largest part of a seed. All this yellow portion is nothing but is the third part, that is called as either endosperm, sorry, not cotyledon, endosperm. Right. Okay. Well, again, there are few special structures which are present in the seed, and I would like to highlight these all structures because many times in many examination it has been asked by some different ways. Okay, so let us discuss one by one. The first important structure, that is endosperm, and regarding this endosperm we have studied also. Okay, so endosperm, what is it? It is a triploid structure. It is a triploid structure which is formed as a result of fusion of one male gamete with polar cell or central cell or polar nucleus. This endosperm is a rich source of nutrition, and it provides nutrients to the embryo during its early development stages. In the previous topic, we have also discussed two things, that on the basis of persistence of this endosperm, in some of the seed, endosperm remains, and in some of the seed, this endosperm is completely consumed. So, on the basis of that, seeds can be either albuminous or it can be non-albuminous. In albuminous seed, the endosperm remains or persists, and in non-albuminous seed, the endosperm is completely consumed by embryo during the development of embryo. So that difference also we will see in our next slide. The next important structure, that is perisperm. We have discussed in the starting of today's topic only that nucellus is acting as a nutritive reservoir. Okay, let me explain like this. And suppose this is the ovary, this is the embryo sac, and this embryo sac is surrounded by nucellus. This is nucellus. This nucellus is again acting as a rich source of nutrient. Okay, so what happened during the development of an embryo and during the development of the endosperm? This nucellus is usually consumed, completely consumed, and hence we are not finding any nucellus in any of the seed structure. But if you take few examples such as black pepper, beet or water lily, if you take the example of these seeds, here the nucellus, the nucellus persists as remnant form, and this remnant nucellus is known as perisperm. If you all have taken the seed of a black pepper, you can easily distinguish it. Today, after completion of this lecture, you can go to your kitchen, take some black pepper seeds and just see. Its outer surface is rough. It is not smooth. On one hand, you can take a bean seed, or in another hand, you can take the black pepper seed. And if you compare, you will find that the black pepper seed is rough externally. It is black color and externally it is rough. It appears something like this. Here you will find there are various types of ridges. This is, then again this is structure, then this is structure, this is structure, and like that. So these ridges are nothing but they are remnant of nucellus. Okay, so nucellus persists as a remnant form, and that is only termed as perisperm. Right? The best example is black pepper, beet, water lily, etc. Okay. Next important part that only I was discussing is integument. Well, talking about the integument means what? External layer. Integument is basically in biteguments ovary. What we have studied, it has two integuments, and these integuments are called as external or outer integument, internal or inner integument. After fertilization, the outer integument converts to testa. The outer integument, this is suppose the outer integument, it becomes very thick, very tough. And this thick and tough part is nothing called as testa. Okay, remember a pea seed. Okay, when we take out the pea seed from its pod, externally you will find a thick coat. Okay, today all those things you can find in your kitchen. So today, just after this lecture, please visit to your kitchen and see all the parts. Because your home kitchen is the best lab. And your mother is a greatest scientist. Yes. Most of the experiments, knowingly or unknowingly, we are doing in our kitchen. And by whom? Your mother only. Okay, so your mom is a greatest scientist. Today, give my regard to all of your parents. So, the external integument that becomes tough, impermeable, so that it can prevent the loss of water from the embryo, and moreover, it can prevent all the external agencies to come and degrade the embryo. So, it acts as a preventive coat that is called as a testa. And the inner integument is called as tegument. So just to know this, in the pea seed ovary you will find a transparent layer. Just the two cotyledons, just before that you will find a transparent, a thick layer that is called as testa, and inner a transparent layer. That inner transparent layer, or inner integument get converts into the tegmen. So testa is the first layer and tegmen is the second layer. This is a very important question again for 1 mark. Okay. Another very important structure which can be found in most of the seeds, and that is nothing but micropyle. Micro, small, pile opening I have discussed many times. Okay, so the micropyle of ovary, the micropyle of gynoecium, that will remain as a small opening. You can see here. This is the small opening. If you take a typical structure of a gram seed, so gram seed we all have seen, it has a small protrusion outside. On the tip, it is roughly triangular, something like this. It is roughly triangular, and this is the protrusion. This protrusion is nothing but it is called as a hilum. And just if you talk about any gram seed, the sprouting of a gram seed always takes place from here only. The sprouting of the gram seed always takes place from here only. Why so? Because there is a small opening, and that small opening is called as micropyle. So these micropiles are the small opening which remains in the seed, and it acts as site for gaseous exchange and water exchange. Okay, so it helpful for the passage of oxygen and water during the germination. Okay, so these are few considerable structures of a seed, right? Well, there are many differences actually, which is mentioned in your NCERT also. And as far as examination point of view is concerned, these all differences are very important. These differences are again very closely related terms also whenever you are pronouncing. So be careful about all the distinguished terms here. So that's why I have listed out few of the important differences. Okay. So the first one is albuminous and non-albuminous seed. This we have discussed in our previous video lecture also. Okay. So albuminous seeds are those in which endosperm is well developed. After the growth of embryo also the endosperm remains, persists, and it is well developed, and hence it is termed as albuminous seed. While on other hand, in non-albuminous seed, endosperm is completely absent. It is completely absent. It is consumed by the developing embryo. Okay. When endosperm is well developed here, then definitely the food storage takes place in the endosperm only, because endosperm is well developed. But then the question arises that you have told that in non-albuminous seed endosperm is completely consumed. Then how the embryonic development stages draw, from where the embryo draw its nutrition during its early development stage? And the answer is cotyledon. Here the food is stored in cotyledon. Okay. For albuminous seed, since endosperm is well developed, hence cotyledon is thin. Much requirement of cotyledon is not important. Okay, because endosperm is very well developed, and that is acting as a rich source of nutrient, reservoir of nutrient. So that nutrient can be supplied during the early stages of growth. But while in non-albuminous seed, when endosperm is completely absent, then here cotyledon will play the prime source of providing nutrition, and hence here cotyledon is very thick. Okay, and but don't confuse here also. Okay, the endosperm. If you talk about endosperm is diploid, it is 3N. If talking about the cotyledon is diploid, it is 2N. Okay, so don't think that endosperm and cotyledon both are homologous. Both of them are acting as a source of food reservoir, but they are not same. Their genotype is different. So at last we can take the example as castor and maize are the albuminous seed and bean and pea are the non-albuminous seed. In my previous video lecture, I also told you either the question can be directly asked that how a albuminous seed and non-albuminous seed both are different from each other, or the question can be modified that how the castor maize is different from bean or pea. So both the differences we should keep in our mind. One is albuminous, another one is non-albuminous. Okay. Well, moving to the another difference. The another very closely related difference is perisperm and endosperm. Okay, because sperm is there in both the terms. And one more difference I would like to include here. Though we have not studied by the time, but you will study in the fruit formation, that is called as pericarp. Okay, so in your notes, maintain the difference of perisperm, endosperm and perisperm versus pericarp also. Pericarp. In the first slide only I told you pericarp covering of fruit. That we will see in coming slides. Okay, so perisperm and endosperm. Perisperm, it is the persistence of nucellus, as I discussed already, that in black pepper the nucellus persists, and that persisted nucellus is nothing but it is the perisperm. Endosperm, it is the nutritive tissue, it is a rich source of, it is a reservoir for the nutrition which is consumed by the early stages of growth and development. Perisperm, since it is a part of an ovary, it is diploid. Okay, it is diploid because perisperm is a part of an ovary, right? And it is a part of a nucellus, and nucellus is diploid, hence perisperm is also remains in diploid condition. But if we talk about endosperm it is triploid. That we know very well. Okay, perisperm, it is papery, thin, wrinkled, shrinked, something like this structure. Okay, and the endosperm we are talking about, it is very fleshy because it is the good source of nutrition. So these are few of the common differences between perisperm and endosperm, and this is your homework. At the end of this video lecture, you will again write the difference between perisperm and pericarp. Okay. Well, now just to gain the idea of various types of seeds, since it is mentioned in your NCERT also, and you all know the changes in CBSE examination pattern which takes place last year, that diagrammatic questions are now emphasized. Okay, so for explaining that, I would like to highlight here few structures of seed which is mentioned here, typically which is mentioned here NCERT. Okay, so one by one we can go through the various structure of seed. Component of seed we are now well aware. Okay, now where these components are present and what is the structure of seed, just have an idea of that. So first of all, I would like to start with the structure of a bean seed. Bean, a very common element, it is present in all the kitchens. Okay, and this is your homework. After finishing this lecture, all this seed you will diagnose, you will see at your home, right? So bean seed, if we are talking about, bean seed is a typical dicot embryo. We know that here. This is the embryo, as you can see, a leafy structure which is present here. This is the embryo. Okay, and this embryo has the upper part, a leafy structure. Here you can find that two leaves are present, like this. Two leaves are present. These two leaves are called as an epicotyle. After epicotyle, this section it is termed as hypocotyle, and the bottom it is termed as radicle. From radicle, we all know it is a root primordia, plumule, which is present here just near the epicotyle. That is called as a shoot primordia and all. The large two structures you are seeing here, from here to here, it is nothing but it is called as cotyledon. Okay, because cotyledon is well developed here, right? And the external covering, the thick external covering which you can find, this thick external covering is called as seed coat, which prevent the embryo. Okay. Next one is seed of castor. Again, let's have a look. Castor seed. This is the external covering. This external covering is called as a seed coat, and it is mentioned here. It is the seed coat. In castor seed, the endosperm remains, and hence this part is called as endosperm. Right? It has cotyledons also. So in the center part, endosperm is external, cotyledon is internal, and this is the cotyledon. This is structure which act as a source of reservoir of nutrition. Okay, and this structure from here to here is called as an embryo, the main embryo. Right. So this embryo, if we talk about, this is the hypocotyle. Hypocotyle, it is the root axis from where the root formation takes place. This hypocotyle is covered by a cap here, which can be seen here. It is called as a root tip of the root, and this is the shoot apical meristem or shoot primordial. Shoot apical meristem or shoot primordial. So this is the embryonic axis. This is the embryonic axis of castor seeds. Okay, let's have a look. Another diagram. A very important and already discussed structure in class 11th. You have elaborately explained this structure or studied this structure of maize, Zea maize, also called as a Zea maize. Okay, so if we talk about this structure, the external covering, it is actually a fruit, right? And externally it is covered by, that is called as a pericarp, that is external covering. And can you name this layer? This layer which is dotted here. Layer, I am not talking about internal. Layer. Yes, that is called as alluron layer. Alluron. It is termed as an alluron layer. Okay, and just alluron layer covers, it is highly nutritive layer. It is rich in protein content. Various typical type of amino acids, they all are present in alluron layer, and this alluron layer circles the largest part from here to here, whatever you are seeing, this whole part from here to here, this whole structure, and this structure is termed as endosperm, or in monocot it is also termed as scutellum. Okay, it is also termed as scutellum, right? One more thing I would like to tell you here, that endosperm is triploid. Alluron also, it is triploid. Okay, alluron is a triploid layer because it is a part which is formed from the endosperm only, right? And this is our embryonic part, right? This structure we have discussed. This is the plumule. This is structure. This structure is plumule. As you can see here, this structure is radicle. As you can see here it is radicle. So from plumule to radicle, this is the embryonic axis. From here to here, it is the embryonic axis. Plumule is covered by the top layer called as coleoptile. Radicle is covered by the bottom layer, that is called as coleorhiza. Okay, so that is a typical structure of a maize seed. Next, a very interesting structure which is there in your book also, that is the seed of an onion, right? So again we can discuss about the structure. The outermost layer, it is the seed coat. This seed coat has, you can see here, like this structure. This structure is called as cotyledon. Right, this U-shaped twisted, like this, a comma-shaped structure, that is called as a cotyledon, and from here to here it is called as embryo. This embryo has shoot apical meristem. This portion is shoot apical meristem. This portion is root tip or root meristem, and from here to here it is called as hypocotyle or root axis. Hypocotyle or root axis. Okay, and very interesting thing, you can see here that endosperm is very small, which is present here. This structure which is present at the bottom, this structure is termed as endosperm. So go through these all diagrams in a very minute way, because again by A, B, C like that they will ask any type of question, and their structures are very important, right? Just try to draw it 2 or 3 times, then you will came to know about all the structures of the seed, right? Well, at the last part of the structure of seed, let's have a little bit look on the germination of seed, that how seed germinates. As far as seed germination is concerned, seed germination takes place by 2 important methods. One is called as epigeal germination and another one is called as hypogeal germination. Okay, so for example, here you can see this is the hypogeal germination. And this is the epigeal germination. So that's why it is called as hypogeal germination and epigeal germination. Right, let us discuss one by one. Most of the dicot, they have hypogeal germination. Hypo means inside the ground. So here the germination takes place inside the ground. Okay, so as you can see here, if you take a seed and sow it, first of all from here you can find few root axes are coming out. These are the radicals which are coming out, right? And here the hypocotyle pushes the root, and at the same time this leafy structure, here you can see this has 2 structures. One is this and another one is here, something like this. It has 2 structures. This is called as cotyledon and this is called as epicotyle. So here the epicotyle comes out first. The first structure which comes out above the ground is epicotyle, and the whole embryo is present inside the soil. This epicotyle take the food from take the nutrition from cotyledon, and then this epicotyle only converts into the first leaf. Epicotyle converts into the first leaf. The first leaf which is coming out from the ground, it is actually not a leaf, but it is the structure that is called as epicotyle structure. Okay. Next one is epigeal germination. Epigeal germination means what? Here the germination of seed takes place above the ground. Here the germination of seed takes place below the ground, and here the germination of seed takes place above the ground. It is commonly seen in the monopod embryo. Okay, so let's have a look about that. So first the radicle came out. This radicle, as the radicle came out, it forms the root axis. When once the root axis is there, this is the hypocotyle region, and this hypocotyle region push this embryo above the ground. As you can see here is the embryo, and this embryo is pushed up. The whole embryo is pushed up along with the cotyledon. Okay, and so roots are developed here. Whatever roots are there, the roots are developed here. The cotyledon comes above the ground, and this cotyledon this cotyledon will remain above the ground, and then it will form the first leaf. Then it will form the shoot pin. Okay, so here the cotyledon and the embryonic structure remains below the ground. That's why it is called as hypogeal germination. Here the cotyledon pushed up the ground, hence it is termed as epigeal germination. Epigeal, hypogeal. Clear? So this is what is two different type of germination which takes place. Well, next, a very important part, and that is called as a seed dormancy. At the starting of this lecture only, I told you that seed can remain in dormant state for long years. Right. So how can we define seed dormancy? Seed dormancy, it is the time between maturation and germination of seed. When once the seed is mature, the time begin from the maturation of the seed. When once the seed is get mature till it germinate, this time gap is called as seed dormancy. Seed dormancy. Remember in class 11th, phytohormones, there are few hormones which are required for breaking seed dormancy. What are they? So that is your homework. Okay, just find it out. So that the time between maturation and germination, that is called as a seed dormancy, right? The variability or viability of the seed can vary from month to many years. It can vary from few months and it can go up to many years. Okay, let me take a very best example here, which is mentioned in your book, right? Some seed remain alive for 100 years. One example I would like to take is lupin, also called as Lupinus arcticus. Lupinus arcticus has the seed dormancy period of 10,000 years. 10,000. What is the basic application of seed formation in angiosperms and why seeds are formed in angiosperms? Every species on this earth has the right to carry forward its generation. Right? And another one more example I would like to highlight here, that recently find out date palm. Date palm can maintain its viability up to 2000 years. Up to 2000 years. Okay, so seed dormancy may vary from few months. Few plants have only few months of seed dormancy, after that it will not germinate. But few plants have 1000 years also. Okay, so these two examples, keep in your mind, it's again very important. So lupin has a seed dormancy of 10,000 years and date palm has a seed dormancy of 2000 years. Okay, right. Well, now come to the fruits. When seed formation is over, when seed formation takes place, seed is now developed. Now next one is our fruit formation. Fruit formation takes place from the ovary. So a ripened ovary is nothing but it is called as fruits. Right? The wall of ovary develops into the pericarp. The external wall of ovary, that wall of ovary develops into the external covering of the fruit, and that covering of fruit is called as pericarp. Okay, pericarp. So remember, nucellus, it was perisperm. For fruits, it is pericarp. And your homework is to write the difference between both of this. Okay, fruit is actually developed from the ovary, but in some of the cases it is not so. So on the basis of that, whether the fruit is developed from the ovary or whether it is developed from any other part of flower, fruit is basically of two types. One is called as true fruits. True fruits are those which develop from ovary, and another one is called as false fruit. False fruits are those which develop from other part of flower except ovary. Other part of flower except ovary. The best example is apple, strawberry. Apple, strawberry, they are the example of pseudo fruits. Pseudo, false fruit. Why they are called as false? Because biologically fruit is a structure which develops from the ovary. And apple and strawberry, it developed from the thalamus. Okay, so it developed from the thalamus part of the ovary. It does not develop from the ovary. And basically, if I am not wrong, it developed from the receptacles. The thalamus has upper part. If you remember the thalamus, this is the thalamus. And this thalamus has the upper part. This upper part is called as receptacles. So it developed from the receptacles. and hence it is called as pseudo fruits. Just as comparing here two structures of a true fruit and of a false fruit. Let us talk. First one is true fruit. So true fruits has three layers: epicarp, external layer; mesocarp, middle layer; and endocarp, which is tough and stony. Endocarp. In mango, it is a season, so whatever you peel out, it is epicarp. The fleshy part, whatever you are eating, is mesocarp, and the inner part, just outside the seed, a waxy coat is present, a little bit tough coat is present, and that is nothing but is called as hard endocarp. Below that, this is the seed. This is the seed. While if we talk about the apple, here the fleshy part, whatever you are eating, it is thalamus. Here the fleshy part, whatever you are eating, it is a mesocarp. Just compare. In true fruits, the fleshy part is mesocarp. In false fruit, the fleshy part is thalamus. So in apple, you are not eating endosperm. Don't write that. What you are eating? You are eating the thalamus. Right. Okay. And this is the calyx. One tip. Here it is the seed. So the pericarp is present. This one. This one is the pericarp. The waxy one. This one is the pericarp. Okay, and in the center of that you will find the seed, as you oftenly find in the apple. Right. So this is a simple comparison between true and false fruits. Well, fruits, on the basis of their classification, whether it form from ovary or not, it is of two types: true fruits or pseudo fruits. But there are various fruits which are developed, which are termed as parthenocarpic fruits. Parthenocarpic fruits or parthenocarpic. Okay. So whether it is in the previous topic what we have studied, whether it is ovary or it is thalamus, it only develops after fertilization. But here what happened in parthenocarpic fruit? What happened here? The fruit developed without fertilization. Without the fertilization, the fruits are developed. Most of the fruits, most of the seedless fruits you could have seen in the market, or you must have eaten also. Grapes are there, banana is there, watermelon. Nowadays, watermelon, it is a season of watermelon, a good source of zinc, which is very important for boosting up your immunity. In COVID-19, we have to take care of our immunity, as most of the time ICMR has issued some guidelines, Government of India, Government of Ayush has issued some guidelines. So go through all those guidelines, very important. So watermelon is a good source of zinc, and that watermelon is nothing but it is a parthenocarpic fruit. In most of the watermelon you will not find seeds. Okay, so like that, right? Parthenocarpy is again of two types. One is called as a natural parthenocarpy, and another one is called as an artificial parthenocarpy. While talking about natural parthenocarpy, the fruit formed as a result of ovary development with pollination but no fertilization. Wow, very interesting. That for the formation of fruit, pollination is mandatory, but fertilization is not mandatory. Okay, and that is called as orchids. The best example is orchids. In orchids, we are developing fruit like that only. Pollination is important, but fertilization is not important. Second one, the fruit formed as a result of fertilization followed by absorption of embryo. So in some time the fertilization takes place. But what is the result of fertilization? What is the end product of sexual reproduction? Embryo. Here the embryo is completely absorbed. What you can find in the grapes. In most of the seedless grapes, the same thing happens. Okay, so these two are the very good example of natural parthenocarpy. While talking about artificial parthenocarpy, here we are performing the same thing by the low concentration of auxin and gibberellin. We are using these two hormones in a low concentration, and this auxin and gibberellin is used for formation of fruit without fertilization. So examples you can see here: seedless oranges are there, seedless watermelon and grapes. All these are the example for parthenocarpic fruits. That is the fruits which are formed without fertilization. When one more term is given, that is two more terms are given in your book, that is apomixis and polyembryony. So let me discuss it. Apomixis, it is a formation of new individuals by asexual method which mimics sexual reproduction including seed formation but do not involve in the fusion of gametes. Apomixis means it is the formation of new individual by using asexual method. But here the sexual reproduction is mimicked, which include seed formation. Formation of seed takes place in this method, but it do not involve the fusion of gamete. Okay, so seed formation takes place in apomixis. It is a very common example of family, grasses family. There we are performing it off. Okay, so what here we are doing, we are taken two haploid cells, which is not the gametes, and that two haploid cells are fused artificially. So here, since two different haploid, it is not called as a fertilization. Typically means what? The fertilization means it is a fusion of gametes, and we are taking other cells, not gametes, and hence this is called as mimic of sexual reproduction. It is mimicking the sexual reproduction, just like the same sexual reproduction steps we are following here, but without gamete. Okay. The another example, the whatever the organism which developed through apomixis is called as apomict. Okay. The last term, that is called as polyembryony. Polyembryony you have seen in various oranges, that is, or various citrus fruit, that is development of many embryo. Poly means many. So development of many embryo, it is a formation of more than one embryo during the development of seed. So more than one embryo is formed, more than one seeds are formed, and that all seeds are termed as polyembryo. Okay. At last of this lecture, let's have a quick look about apomixis and polyembryony. So apomixis, an asexual reproduction that occurs without fertilization, but it involves and it do not involve meiosis. While in polyembryony, the formation of more than one embryo from single fertilized ovum, more than one embryo from single fertilized ovum. Here fertilization is taking place. Here no fertilization. Right? Second one is, it is a typical asexual type of reproduction, though it mimics like a sexual reproduction. But here it is a type of a sexual reproduction. We are developing many embryos by multiple fertilization. Fertilization do not occur in apomixis, and here fertilization is mandatory process as we have discussed. It does not involve the production of seeds. No seeds are formed here because no fertilization takes place. And here seed production takes place because fertilization took place. And last one, the offspring of plants are genetically identical since it is a asexual mode of reproduction. So all the offsprings are genetically identical, and here the offsprings are not genetically identical. Right? So this is all about the whole topic. Today we have discussed about seed formation and fruit formation. We have discussed about various comparative structures of seed and fruit. We have also discussed about the development of seed, epigeal or hypogeal. We have discussed about structure of various seeds. We have discussed about the structure of fruit, true fruit and false fruit. And we have discussed about apomixis and polyembryony, including parthenocarpic fruits. Friends, by this lecture, this is the 6th lecture of your second chapter of 6th unit. Hence, this chapter is now over. We have completed, we have discussed elaborately about this chapter. Definitely, many doubts must be there and doubts should be there when you read the chapter, keep on asking your doubts with your teacher, right? By the time, take care of yourself, keep on revising. Next time we will do something different. What is different? That you will came to know through your class groups and by your respective teachers. But in next video lecture, something we are going to do different. One small hint for that: study this chapter very thoroughly, and let us see who is the winner of next lecture. Next lecture will be taken by you and studied by me. Take care. Bye bye.